Today topic of discussion is passive transport. Transport across the cell membrane are divided into three main types: active transport, passive transport and vascular transport. In this video we will discuss about passive transport. What is the meaning of passive transport? Transport of molecules across the membrane down the concentration gradient. Okay, what is the meaning of down the concentration gradient? From higher concentration to lower concentration. In this method, energy is not required. So, what is passive transport? Movement of substance down the concentration gradient. In this method, energy is not required. Then, four type of passive transport. What are the four types? Diffusion, simple diffusion method, fascilated diffusion method, osmosis and filtration. First we will discuss about simple diffusion. Transport of molecule down the concentration gradient, that means higher concentration to lower concentration. In this method energy is not required and carrier protein also not required. So simple diffusion means 3 points is important. What are the 3 points? Transport of molecule down the concentration gradient from higher concentration to lower concentration. Second one is no need for energy and carrier protein. Now there are two mechanisms of transport in the simple diffusion. How many? Two mechanisms. First one is transport of lipid soluble component and transport of ion. First, transport of lipid soluble component, mainly urea and oxygen gases. Okay, it transported through lipid bilayer. Urea and oxygen gases transported through lipid bilayer because it is soluble in liquid. It is travel to the membrane. So transport of urea or gases oxygen from higher concentration to lower concentration across the membrane because it is soluble in lipid. This is the first mechanism of simple diffusion. Second, through ion channel. This one is the black color one is the ion channel. Through ion channel transport of ion occur because of charge. The ion cannot transport to lipid layer. Because of that charge, that molecule cannot transport to the lipid layer. Okay, by using this channel, this acts as a channel. By using this channel only, this ion molecule travel from higher concentration to lower concentration. Number of example of ion channel: sodium channel, potassium channel, calcium channel and chloride channel. There are two types of ion channels: voltage gated and ligand gated. What is voltage gated? Means opening and closing of the channel are dependent on charge developed across the membrane. Okay, based on the charge, sodium, calcium, potassium, that opening and closing of the channel are depending on the charge across the membrane. It is called as voltage gated. Second one is ligand gated. What is ligand gated? Means opening and closing depending on their chemicals are known as ligand gated. The main ligand are acetylcholine and glutamate. Next one is clinical significance. First one is chloride channel. There is mutation in chloride channel, defect in the chloride channel which causes cystic fibrosis or fibrosis. Okay, it leads to Mutation in chloride channel, it leads to cystic fibrosis or fibrosis. Various drugs which are used for the treatment of hypertension or diabetes mellitus affected the ion channel. Example, sulfonate urease. So what are the clinical significance? Mutation in chloride channel, it leads to cystic fibrosis. Various drugs which are used for the treatment of hypertension and diabetic mellitus, it affected the ion channel. Next one is second method, fascilated diffusion method. The only difference between simple and fascilated diffusion is carrier protein. It is a carrier mediator. Transport it down the concentration gradient. That means higher concentration to lower concentration. Energy is not required, but carrier protein is required. So what are the 3 points? Down the concentration gradient, higher concentration to lower concentration, energy is not required, but fascilated diffusion required carrier protein. This method follow ping pong mechanism. This one accepting state, this one is the pong state, releasing one, this one is the ping state. It follow pong state, that means accepting mode. The molecule to be transported crossing the membrane, it is attached on the external part. Okay, this one is attached to the external part of the carrier protein. When it attached the external part, the carrier protein, the conformational changes in the protein will occur. The conformational changes in protein occur due to which the molecule get internalized. So it is the transport down the concentration gradient, mean from higher concentration to lower concentration. Via carrier mediator protein, energy is not required for this process. When the fascilated diffusion occur, when it across the membrane of the one molecules only one molecule transport across the membrane, it is known as uniport. Transport of the single molecules across the membrane name is called as uniport. When the two molecules are involved and they are transported in opposite direction, chloride inside, bicarbonate to outside. Okay, opposite direction, it is known as antiport. Example of uniport diffusion: absorption of fructose from intestine and transport of glucose in skeletal muscles and adipose tissue. It is a insulin mediated transport of glucose in skeletal muscles and adipose tissue. The examples of antiport: two molecules. What is antiport? Two molecules transported in opposite direction. Chloride bicarbonate exchanger, which is presented in gastrointestinal system in the stomach. It is presented in stomach and it is responsible for HCl secretion. Second one is bicarbonate chloride shift. Chloride shift in RBC. These are the two examples of antiport. Chloride bicarbonate in gastrointestinal system. Second one is bicarbonate chloride shift. Third method is osmosis. What is osmosis? Solvent. Solvent means water molecules move through a semi-permeable membrane. Okay, with the help of semi-permeable membrane from a diluted solution to more concentrated solution, lower concentration to higher concentration solution. The water molecule will move through semi-permeable membrane from diluted low concentration to higher concentration. Here solvent is water. What is the clinical Significance of importance of osmosis. Edema. First one is edema. Edema occurs whenever the albumin concentration decreases. It causes necrotic symptom and liver cirrhosis. For this, we will give 0.9% normal saline in folate loss. It is a isotonic saline. When we give that normal saline, RBC hemolysis does not occur because of isotonic solution. Fourth method is filtration. It is a mechanical pressure gradient which force the fluid to flow from higher pressure area to lower pressure area. The pressure development in the body, it due to cardiac activity or its cardiac output, then gravity, then vascular tone. It is responsible for development of higher pressure. It is important in transport of nutrition and water to the tissue via capillaries. Next one is important for glomerular filtration. Help in the urea formation, means removal of waste from body via the kidney. This is all about passive transport. Notes format: passive transport, down the concentration gradient, 4 types: diffusion, fascilated diffusion, osmosis, filtration. First is simple diffusion. It have 2 mechanism: lipid soluble, then transport of ions channel by using ion channel. Two type of ion channel: voltage gated, ligand gated. Then clinical significance of simple diffusion, then fascilated diffusion. It follow ping pong mechanism, uniport, antiport, along with the example. Then osmosis, filtration. Thanks for watching.